because, well, people pay me to study cell migration because that is how you understand why cancer is bad. And there's one thing that you must remember, because if you look at a microscope, cells randomly migrating seem to go quite a long distance. But on macroscopic distances, like your bloodstream, etc., random migration is useless. The only time cancer will spread is if it migrates somewhere, there is something steering it. And in quite a lot of cancers, it's a chemical that is steering it, so it's chemotaxis. So that's why people pay me to study that in a cancer institute. I'm moving to a much more university-based background. And they want me for a completely different reason, for exactly the same research, which is information. Why is it that cells do what they do? How do you understand the patterns of cells that you see in organisms? And again, there's lots of chemotaxis involved. Let me express this as clearly as I possibly can. That's what you looked like a while ago. In order to get from that to that, quite a lot of chemotaxis is involved. Obviously, this thing doesn't have much shape in it. And quite a lot of the cells that give shape to one of those, the way they get their shape if the cells migrate up gradients. But it would be stupid, it would be completely dumb to say that there are gradients built into this thing that inform the cells where to go. The embryo has got to create its own gradients. So there is a lot of cell migration involved, but there's also generation of the complexity and generation of the information involved in it. Two interesting things, complete sideshows. First one is I am probably the only person you'll ever met who's seen his own twin children as full cell embryos. They were test tube babies. The first round of test tube babies we had, I said, I would be really interested in seeing what the embryos look like. And they said, you cannot do that, it's illegal, it's evil, we will prosecute you, we will, you know. Second time I did it, I said, I am a professional microscopist and I do not think the gear you are using is very good. And they had me in the room in a second. And here we are, look, Insall, here we are, look, here we are. So I saw my own thing. Second thing is, I'm very modern and do not go showing pictures of anyone's children, even my own, in talks. That is generated by a GAN article. We have a whole program using GANs to illustrate cancer. If you're interested, give me an email or read our account. The one thing we do not like studying is the cells chemotaxing along a pre-existing gradient. Because if you are studying cells chemotaxing along a pre-existing gradient, then you're just kicking the ball up the field. There's nothing, the cells are taking the information that you are feeding them and they're interpreting it to give the result that you fed them. It's not interesting. What we're interested in is where the complexity and the information come from and how the chemotaxis generates it all. And if you want a nice person, Bob May, who is an utterly brilliant biological statistician who informed the government and so forth in years gone by, said if the system doesn't organise itself, then who is going to organise it for you? Okay, and here is a nice picture from 1952 or 3, I think, before my time, of a neutrophil chemotaxing towards a bacterium. And this is by far the most well-shown and most appreciated movie. And you can see on the one hand, it's incredible, it's so anthropomorphic. Mm-hmm. You can see the bacterium saying, and the neutrophil chasing after it. And on the other hand, this is really boring because the information is always coming from the bacterium and it's not going to change much. And we're interested in where the information comes from and how it's understood. Okay, let us jump from here to cancer, because that's where we're at. Melanoma spreads by chemotaxis. We know melanoma spreads by chemotaxis. It's pretty clear melanoma spreads by chemotaxis. One of the ways you can tell, since we're in a mathematical institute, is that a melanoma is not a Gaussian shape. It has complicated shapes and it has sharp edges and so on and so forth. If the cells were just growing and dividing and moving randomly, the melanoma would be Gaussian. And the further it is departed from Gaussian, the more frightened your physician will be by it, because that is the definition of a bad melanoma. It's not rocket science. Here is a nice assay along the lines of what I showed you. So these are devices that I build, which is to say I build them in Blender and then I pay someone to make them. But we have a well here, we have a well here, and we have a band 20 microns high, which is as long as I wish it to be, which is why it's nice to build your own devices. And a diffusional gradient is set up between here and here. Sorry, I've got a pointer and people then see it. A diffusional gradient is set up between here and here. And it's linear. And I should say, sorry, let me, normally what you do is you put the cells everywhere on this bridge and you try and expose them to a simple gradient between low and high. But we worked out a while ago that it was much more interesting if you just put the cells at one end and watch what happened. And you'll see why in a bit. Anyhow, here are melanoma cells moving from low serum to high serum. And it's brilliant. It's very, very beautiful. It's also absolutely terrifying. You should be quaking in your boots when you see this, because this is about the distance from the median cell in a melanoma to the lumen of a capillary nearby. And what if you had a melanoma and this was happening, then you would be going to die of it, absent therapy.
And the gradient's linear.
Oh, it isn't. It isn't in here too. How do we know it is in the device with no cells in it? Because it's one-dimensional. We can verify it by putting fluorescent things in, and we do, but it's designed so it can't be anything else. But later on in the talk, you will find that the gradient is very far from in here. So very good question.
Similar question about the protocol.
Yeah.
So exactly what is the system? I'm just curious.
What if the chemicals and the cells?
No, like how do you create the gradient and what kind of
So you have a well that is 1 millimeter deep here, and you have a well that is 1 millimeter deep here. And to a first approximation, this is infinite in the y directions. That's why it's one-dimensional. And then you have a 20 micron gap between the two of them. Sorry, I should be pointing with here, shouldn't I? So between here and here is only 20 microns high, so there's no flow. The viscosity is stupidly high in this direction, and the flow is fairly easy in this direction. So between, you can see the edges of it by the microscope artifact. Between there and there, if it's set up right, everything is set up by diffusion. And it takes about, depending on the size of the molecule, it takes 10 minutes to diffuse into a nice linear gradient. I will show later on a model of a gradient forming in one of these. But the person who set this all up was an absolute genius called Sally Zygmunt in about the 1950s, 60s. And one of the things I realized very recently is that one of the reasons Sally Zygmunt was a genius also totally screwed up the field, because she set up something where it was safe to ignore all the things the cells were doing to their environment. And you could only see the change that you were imposing on them. So it made it possible to study neutrophils, and that was great. But it made people think that neutrophil behavior was simple. And if we have learned one thing, it's that almost no chemotaxic cell's behavior is simple, and neutrophils' behavior is complicated to the nth power exponentially with oak leaves and extra cells. So there's the device, but you can just imagine that between here and here there is no liquid flow, but there is a diffusional gradient. And you see another interesting thing, which I will explain in a couple of slides, which is that the cells don't move consistently, they move in a band. And what happens when you set it up the way Sally Zygmunt described it, if the cells all behave consistently, so that you can draw consistent conclusions, and this is a complete illusion. What you're seeing is unbelievably complicated, but it's a nice rendering of it in her essays. Anyhow.
You have a couple of cells sitting in the middle of your pattern
Yes.
doing absolutely nothing.
Yeah, stochasticity, isn't it wonderful?
Screws up everything.
What you can see though, hang on, come back. What you can see is that when the cells get to the middle, they don't have very much steering, but when the steered wave goes over them, they acquire steering, which is interesting. Okay, oh gosh, I'm pressing the wrong button. Concentrating on the questions, not my system. Sorry. Okay, so the question is, if cells in a melanoma are chemotaxing from the melanoma to somewhere new, they need information. Something has to be defining what is melanoma and what is hue. Let me word this differently. Why does your body create gradients in order for melanomas to kill you? It seems a very stupid thing to do. And the answer, of course, is your body does not create gradients for melanomas to kill you. Melanomas create gradients for melanomas to kill you. Your body is not really that much involved in it. And the scary way we proved this, and it was scary because the first time we did it, it was a control. But thank goodness we're a mathematical lab. So when our controls were wacky, we went and tried to explain it. Here we have no gradient. Here we have high serum at this side of the gradient, high serum at this side of the gradient, high serum here, but the cells are only in this side, and there's no difference. And there's so much no difference that for the first year or so I was speaking about this, I got the sides switched and didn't notice. Okay, so what's actually going on here? What's going on here is that these melanoma cells are creating the chemotactic gradient that they are chemotaxing at. In serum, there is a whole load of chemoattractants. The reason you use serum in biological experiments is serum is this chicken soup of every attractant and every growth factor and every stimulus that you can find in the bloodstream when it's all turned on. And so what these cells are doing is they're seeing a saturating stimulus here, and here they have broken down all the stimulus. And between the front end and the back end of this wave, that's where the gradient is, and the wave of cells is making it as they go, which is cool, and which is my way into this field, really. And it turns out, this about 6 or 7 person years of work, that the chemoattractant is a blood cell chemoattractant, mostly called LPA, lysophosphatidylate acid. And it turns out that these cells are amazingly good at breaking down lysophosphatidylate acid. So if you sit cells in clean medium, they don't make lysophosphatidylate acid. But if you put a low density of cells, shown here in blue, then they break down all of the lysophosphatidylate acid that is in the serum over a little while. And if the cells are confluent, they take out all the LPA in a day or less. So another thing that we have found more recently is that this is not the real chemoattractant for melanomas in you. This is the chemoattractant for cultured melanomas because they've been cultured in serum and it's full of LPA. And we think we know what the chemoattractant is for actual melanoma cells, but maybe there's several. Is it
generalizable to any sort of assays we're using serum for gradient for the
amazingly
migration?
so, yes.
Oh.
Appallingly so. And it's not just general, I mean, LPA, as well as being a chemoattractant, is also an amazing growth promoter. It really does cause mitogenesis. And so when you feed cultured cells, they get this great shot of LPA that they break down in some number of hours. So you think that you're just feeding your cells and keeping them all right. But actually what you're doing is giving them a discrete, saturating growth stimulus that they will then break down. And it's weird considering all the things. So you'll be getting the message from this. I'm a biologist that's been converted into math. And the more I do think like this, the more I realize we have been just doing things that are complicated, and thinking they're just run-of-the-mill. Very interesting. Okay, anyhow, so what we've got here is self-generated gradients. The cells are generating the gradients that they are responding to. And it turns out, as we study more and more, that just about every gradient is influenced, at least to a degree, made by the cells.
Is LPA, by the way, a lipid species?
LPA is a fairly soluble lipid. Yes, it's a charged head group and a tail. Well, it's a soap, if you really want to think about it like that. It's so it's a tiresome to work with, and in fact, most of the time we work with it now. We work with serum plus or minus a very good inhibitor of LPA receptors, because serum is very good, it's stabilized, it's soluble, whereas if you stick a pipette in LPA, then the pipette's all coated with it and you have no idea what comes out into your sample.
And is it used for the membrane generation
No,
or energy?
it works at 1 nanomolar. So it's irrelevant to membranes. It's a signaling molecule that comes from lipids.
Signaling.
Yeah, rather than yes. Okay.
So if we constantly pump in LPA, you would not get directed migration.
That's right. If you could do it constantly enough.
If you could do it
Yeah.
constantly.
And in fact, that's true for almost every chemotaxis experiment everyone does. If you consistently put it in, the results would be completely different from what you measure. Interesting. And remember, as I keep talking, all of the information is coming from the cells in self-generated gradients, not from me. Okay, how are we going to understand this? If we go and do experiments, we will do positive controls and the cells will still work in them and we will not understand anything. You can't understand this sort of thing by experiments alone. It's not good enough. So we make computational mathematical models. Actually, nowadays, we do quite a lot of analytical math models because you can express the breakdown of chemoattractant in space as a PDE. And so it turns out that we now understand this system well enough for some cells that our analytical math models are right, which makes me incredibly happy and leaves most of the biologists cold, which is okay. So again, what we get is spatiotemporal complexity, we get simplicity, and because we have systems of PDEs and positive feedback, we can say these two things in the same mouthful. My wife, who is a very senior and very excellent biologist, when she sees complexity, says, this is no good, we're going to have to get rid of it because human cognition is not good enough to understand it. And I say, nonsense, use equations. And what we're getting, because we have analytical math, is we have interpretability and explanation, and we know that our explanations are right, so it's cool. What we don't like is linear pathways. Thank you very much. And we are constantly being asked by editors, could you please write a pathway diagram to show what's happening? And we constantly say, no, that makes you think what is happening is a linear pathway, but there aren't linear pathways in this system. And it's a math biologist's paradise. You've got positive and negative feedback. You've got instabilities. I'll show you a hop bifurcation later on.
You've got more than one factor that's influencing that.
That's right.
LPA is presumably a dominant one, but it's not the only one.
In those melanomas that you saw before, LPA is overwhelmingly dominant. I think. Oh no, I took the slide out because I'm going to go over time because you're asking questions. Yeah, no, that's absolutely right.
You're allowed to go over time here.
In some conditions, there is one overwhelmingly dominant thing. With the dictyostelium, which is why I still work on them, even though they're very biologically unfashionable, there is only one receptor. You can ignore all of the other ones. With, interestingly, dendritic cells, there are only two receptors that matter very much. It's still more complicated, but dendritic cells are modelable. With neutrophils, just forget it. I mean, just don't even try. There is so much going on that we're going to have to keep using descriptions on them.
So I guess we know cancer cells, for example, are producing growth factors, matrix proteins, matrix degrading proteins.
Yes.
All sorts of stuff.
My guess is that in most cancers, all of the stuff you have to consider all at once. And my guess is that in some cancers, and melanoma is one of them, actually you can ignore nearly all of the stuff. So I mean, with melanomas, people got very, very worried about basement membranes. And whether the cell can penetrate the basement membrane. And it turns out that if you look at a mole, so totally non-malignant, it turns out that the basement membrane is annihilated for centimeters in either direction. So that whether the cells break down the basement membrane in melanoma is really not terribly interesting. Which is not to say it's not interesting in any other tumour, just that it is safe perhaps to ignore it in that one. Okay, so here's the first model I'm going to show. This is a kind of really nice, simple agent-based model, but it's the best way of illustrating what's going on that we can see. There is a chemoattractant. The cells break it down, so that's the gradient caused by the cells breaking it down. The cells migrate in a wave. Some of the cells migrate in this wave. A number of cells are left behind. Why are cells left behind? Because we actually showed this with very interesting analytical math. The last cell in the wave is seeing almost no information. And if the wave gets a little bit ahead of it, then it's got zero information, so it doesn't follow anymore. So all these cells back at the rear, they're all there because of chemotaxis. If there wasn't chemotaxis, they wouldn't be there, but they're not chemotaxing when you look at them. It's very interesting. If there were not enough cells in that wave, then chemoattractant would leak through, and then more cells would be recruited to that wave till there was exactly enough. So there is an analytically correct number of cells in a way. Interesting. So masses of attractant here, much more than the cells can perceive if they're not self-generating a gradient. And it turns out that in about half or maybe 2/3 of all experiments the wet biologists do, us wet biologists do on chemotaxis, we put something like 100 times too much attractant in, so the cells should never have been able to read the gradient, but they do, because they weren't reading the gradient, they were making the gradient.
All the cells are the same to each other, right?
All the cells
Are the same.
in this model, yes.
There's no heterogeneity.
In different cell populations, more and more. Macrophages, for example, there's no such thing as a homogeneous population, and you can see them all sort of spread out during assays. In this case, they're all the same.
Yeah.
And there's lovely math varying different parameters in subgroups of cells.
I was wondering, looks interesting, like at the end of the video, it almost looks like, I mean, I can't tell, like almost like, I don't know if a Gaussian, because I would expect it to be uniform on the left, but it doesn't look like that.
It's far from that. And actually, it's a complicated thing because the velocities, maybe I'll show it. Very cool question, actually. The reason the wave is arbitrarily stable over time. And we've actually shown that we've measured this with real cells. So the velocity of the cells just at the back of the wave is much higher than the velocity of the cells in the middle of the wave, which is how the wave is self-focusing. So the cells at the front of the wave are just entering a domain where their receptors are saturated and they have very, very little information. So they have a same speed but a lower velocity. And the cells right at the back either have peak velocity up there or 1 instant later they have no information left and so they're lost. And you can keep these assays going for days and the wave stays the same profile, even though you would expect it to be spreading out by randomness, but it's self-sharpening. Those of you who are biologists, it's exactly the same mechanism as a Lemley protein gel. It's self-sharpening leading edge.
It looks like behind your leading front, you allow for cell proliferation.
Yeah.
In this model, yes.
Yeah, and then you get your beautiful Gaussian
That's
distribution of the population behind it.
That's right. Yes. Not quite Gaussian, but it's yes.
Well uniform.
It's a gamma curve of some sort. Okay. Why does melanoma like self-generated gradients? You have to think back to when you were an embryo. And when you were an embryo, you were unpigmented. Every one of you, and this mouse as well. You can see it. And actually, you sort of know this in the back of your mind. If ever you see an embryo of any kind, they're always sort of white and uncovered. So when you are an embryo, your pigment cells emerge from your neural crest, and they migrate from the neural crest through the dermis all the way around, and all the way around, and then down your arms. If they don't do that, then you end up with patches, especially in this axis here, that are unpigmented. And actually, there are a number of syndromes where people have, there's one pair of syndromes where people have a white triangle in the middle of the forehead, for example. It's very, very diagnostic because the melanoblasts do not move correctly. And amusingly, Laura, my wife, had a mouse colony and she was looking at RAC signaling in them, but in that melanoblasts, and they all end up with white bellies. And so we ended up simultaneously working on the same thing just by accident, because she had all these mice with white bellies and I had these models. Anyhow, I'm talking too much. This is an astounding experiment that we did. I've not been able to do anything like it, where we have labeled in vivo the melanoblasts in an embryonic mouse and watching the confocal microscope. And you are looking at the shoulder of the mouse here. And so that end is the paw end and that end is the body end. And you're looking at the melanoblasts migrating from the melanoblast-free area that was started at the neural crest to the end. And you see this gorgeous movement of those cells. They are the most beautiful cells, but the ones at the front are directed. You can actually see fairly clearly they're moving on the whole down the arm. The ones 400 micrometers behind are not, they're moving randomly. So the simplest explanation of this is that when your pigment cells migrate, they are migrating to a self-generated gradient that takes them from where they are not, where they are, which is the neural crest, to where they are not, which is all the way down to your fingers. And the reason melanoma is so spready is that all you need to do to a melanoma is just de-differentiate it a bit. And it thinks it has to do two things then. One is it has to one is it has to proliferate.
Yeah.
And the other thing it has to do is spread out from where it currently is to somewhere where there are not cells, which could be a definition of metastasis, if you want. And I didn't add the extra slide that I promised. I'll have to show it to you later, Sandy. Yeah, later I will. But it turns out that the exact thickness in the plane going into your skin is by far the most information-rich diagnostic of whether a melanoma has spread or not.
Is it biologically more clever to generate a gradient out of an existing material or to release a chemorepellent?
Very, very, very interesting question. The answer was wrongly created by Hans Meinhardt. If you know Hans Meinhardt, he's a very brilliant biologist of this. The reason he got it wrong is he thought about a chemoattractant as a fully continuous stimulus, but chemoattractants are interpreted by receptors, and receptors saturate at one end, and you don't have any receptors occupied at the other end. So if that lovely thing I showed you with the wave is self-sharpening with a broken down chemoattractant because the cells at the front of the wave saturate and the cells in the middle of the wave accelerate, or at the back of the wave accelerate towards the front. If you do the same thing with a chemorepellent, the cells at the back of the wave are saturated, so lose their velocity. The cells at the front get the most. So a chemorepellent is dispersive.
Yes,
And a chemoattractant is focused.
Interesting.
that's right.
Either of them work brilliantly, and there are biological instances. It turned out that I was working on this a full decade before I knew I was, because Entamoeba, amoebic dysentery, horrible, horrible disease, kills lots more people than almost all the diseases that medics study, is driven by a self-produced chemorepellent. And we found this in about 1990, but I didn't have mathematicians. I hadn't transitioned at that point, so I couldn't understand the breadth of what they were meaning. But yes, so both happened.
I have a question now. Are you saying like these cancerous melanocytes or melanoma cells that they want to go, because they're de-differentiated, they want to go from where they are to where they're not?
Right.
I'm wondering, would they be able to, can they, I don't know, anthropomorphic, can they tell the difference between then what they are as de-differentiated versus differentiated
No,
melanoma?
probably not. No,
melanocytes?
probably not. So we have a model that I'm trying to publish, but it's turned into this huge thing, showing what is going on in the embryos, and we can perhaps answer that question in the model, which is that we think that actually during the embryos they are doing two different self-generated gradients. One which goes from the neural crest to the ends of the fingers, but the other which goes from the dermis into the epidermis. And normally what the cells do is that they do the first one first, and then when they run out of that stimulus, then the upward one becomes dominant. But in order to be a melanoma, you have to have overcome the need for growth factors because there isn't a supply of them. So this other pathway is not active. So they are stuck, perhaps, in the idea of being melanoblasts rather than melanocytes, which are the kind that move up.
Okay, I see.
All good questions. All excellent questions. Go on.
Is the knockout model of the receptor made, and if it is known, is the receptor
The
used?
knockout model is really tough because it controls growth and differentiation as well as chemotaxis. And this is actually generally true. And I have a whole nother talk on this if you have another hour spare time. But it turns out that these things are very often coupled in biology, and they're coupled for a reason. But the knockout model is no good. But for LPA, there is an extremely good competitive inhibitor of the dominant receptor, and it just wipes it out. It's brilliant. So you have cells that are the right identity and in the right place, and you add this inhibitor, and they don't go anywhere. And the physicians in the audience always then want me to put that inhibitor into patients. And the answer is, A, you'd stop all your blood cells from proliferating, and et cetera, the patient would get very sick. But B, this system is a positive feedback loop. It's insanely robust. And drugs can typically take out 75% of the activity of a pathway. Taking out 75% of the activity in these videos I've shown you means the pathway still works, but you get 4 times the number of cells in the wave. So not helping the patients very much at all. But it's an interesting little world.
Maybe I misunderstood. I thought you mentioned that LPA is relevant for these in vitro experiments, but
That's right.
implication
Yes.
was there something?
Yeah. We discovered that actually lately, which is why I'm mixing up the timeline like that. There's another reason you wouldn't give people drugs for this. Yes, it's not doing it.
But I'm curious, like what it is in vivo that acts as an attractant.
So the embryogenic attractant is endothelin-3. And the mutations that I told you that have white spots on their forehead are incompletely penetrant mutations in the endothelin-3 system. The attractant for actual melanomas, we don't know. We discovered after we stopped working on this that it wasn't LPA. And it would be very interesting to go back and take an informatic approach to find out what is. But I haven't got around to it. There's so much left to discover in this field. It's brilliant. Okay, I'm going to tell you a little bit about why cells would use these things. So we're getting into a much more sophisticated model here. This is all based on PDEs of interacting PDEs of the attractant diffusing, getting broken down by the cells. So there's an agent-based layer with the cells in, but the cells are just using PDEs here. And that is what you normally think happens. So the chemoattractant diffuses across, the cells interpret it. And if you think about it, at the low end of the gradient, the cells have got attractant at the front and zero attractant at the rear, so they have a very good grade of information. But here, 2 bad things happen. One is they've got attractant at the front and a bit less attractant at the rear, so they've got less information. And the second thing is they start getting saturated. A gradient that is simple is not robust. If you look at a self-generated gradient, it's unbelievably robust because for all the cells, there is attractant in the front and there is not attractant at the back because they've broken it down. So it is, as I've said, it's robust for an arbitrary distance. And actually, this means that simple chemotaxis, where something makes the attractant and the cells just respond to it, have a terribly limited range. You think about it, if you give cells a low concentration of the attractant, I'm showing the concentration that cells can perceive here in green. You give them a low concentration, initially they can perceive it, but it spreads out into a very flat gradient, they can't see it. So, okay, let's give them a high concentration. So then you get a lovely domain that is interpretable, but by the time the cells have moved into that, they're saturated ahead of themselves and they can't go very far. So you cannot ever migrate 5 millimeters to simple chemotaxis. There is not enough information in the world. But in a self-generated gradient, the gradient is only where the cells at the leading wave are. And there's no information before, and there's no information behind, and there's no occupancy behind, and there's too much in the front. So it's beautiful. You've got wonderful information for the cells that are in the wave, and it's terribly robust. Yes?
How long would the channel have to be for like a second pulse to generate?
It's a different. It's a good question, but different. There's no source of attractant in this system, so there can't ever be a second pulse because they've broken it down. But if you have a more complex system, and we have modeled this, of course, where there is a constant input of, for example, you don't get a second pulse. What you get is these cells. Oh, stop pressing buttons. Sorry. Oh, I'm a bad man. There we are. What you get if you have a constant flux of attractant is that this pattern at the rear spreads out. And the cells go to a non-random spacing from one another because each of the cells is breaking down. And of course, if you keep changing the scenario, you'll get a different result each time. So if there was someone else who was making an attractant, then you could find a condition where you'd get a second pulse.
Okay.
And this is robust to all parameterizations of the model, or
There
must be some differences, right?
There's an infinite. No. There's the models. So we have reached the point where our models are such a good representation of reality that when reality and the models differ, it is because there is a thing that the cells are doing biologically that we didn't know before. We found a number of things, of bits of new biology. Actually, Dennis Bray, who did the first models of bacterial chemotaxis, his models got so good that if the models were wrong, it was because the biologists had messed up their measurements, which is brilliant.
The model itself is just agents, they're all the same. They secrete a signal, I suppose, that this model is a PDE.
No, in this model, they just break it down. Dictyostelium, so that's one of the things we found that was wrong, actually. Yeah. You hit the nail on the head. Dictyostelium are capable of secreting signals. We had put a drug on them to stop them.
Yeah.
And the cells did not agree with the model. And it turned out that the drug was working 95% of the time, and the remaining 5% made the cells behave differently.
Yeah.
So we knocked out the gene, and the data I've shown you are mostly with the gene knockout, which is 100%.
Yeah.
That is a consistent. This is the wonderful thing about modeling. Once your modeling is good enough, and actually some of your oncological models are good enough. Most people's aren't. But you've got a unit here that works on it. You've been improving, refining, so on and so forth. I'm not insulting. It's reality. If you work on something properly and try and improve it, then it gets good.
But one more question is, how many agents do you need to get a robust invasion? Because I assume if you just put one cell in
It's
there.
totally parameter dependent. We've got, I am waiting, this is such a shame. We have a beautiful paper showing that one cell can make its own attractant, its own attractant gradient.
It can, but then it can go up and down as well, right?
That's right. So you get autochemotaxis in a random direction.
Yes,
Isn't that
right.
cool?
Yes.
So that's why I say, how many would one need to get this
I
Direct
see.
it.
10.
10.
Yes.
You make that up as you go along.
5, you get stochasticity, and we've done 5. But the trouble with, you have an interesting thing which someone will ask me about, so I'll answer it now, which is that the system we're describing has a wavelength.
Mm.
And in the cells I'm showing, they're very, very rapidly moving, and the attractant is very diffusible, so wavelength's huge. But in cancers, the wavelength is probably, so what you see with cancers is probably you see fingers coming out because the wavelength will give you a finger instability.
Very cool,
I
thanks.
guess adhesion would make a difference there too.
Yes. Yes, and those cells, like fibroblasts that are mostly defined by adhesion, it would make 100%, the adhesion would change everything.
And they are breaking the attractant like 100%, right? There is nothing left.
What the analytical math says is that the number of cells you will see in the wave will be enough to break down 100% of the and if the only thing that is not stable is if you don't put enough cells in the assay. And if you don't put enough cells in the assay to form a wave that breaks down the complete flux of attractant, then the experiment doesn't work properly. But you can put 10 times too many cells in, no problem at all, they just get left behind. 100 times too many cells, also no problem, but slightly too few to make a wave messes the experiment up. Oh, I didn't say. So what this means is, if you ever look at anyone talking about chemotaxis, if the gradient is less than 0.5 millimeter, it could be a self-generated gradient or it could be a passive gradient. But if it's more than about 0.5 millimeter, certainly more than 1 millimeter, it cannot possibly be a passive gradient, it won't work. In embryology, for example, and quite a lot in cancer, people are talking about gradients that are often more than 1 millimeter, so they must be being made by the cells, at least in part. Okay. Saying we extend this model a bit and make the cells make a decision. So we're going to put cells here, and they can go left or right. And I'm afraid I had to take out quite a lot of the interesting slides here, but we had lots of fun with this idea. This one I'm showing you, in our models, blue means connected to the outside world. So blue has a source of fresh attractant. So now the cell is deciding between two similar lanes, but one of them has a source of attractant. And what you see, if the cells split two ways, but then the lower tract gets replenished, so more cells end up going there. That's pretty much what you'd expect.
Yeah.
Right. Okay. Now I'm going to make the unconnected lane a bit shorter. And now, by the time the cells arrive, there's actually a difference between the top and the bottom channel that they've made by breaking down the attractant. So actually almost no cells go in the top one, because by breaking down the attractant, they have been able to tell that the upper channel is not connected to the outside world and the lower one is. So let's carry this to its logical extreme and make a maze. And if you make a maze, the cells are perfect. The modeled cells are perfect, because by the time they reach the decision point, they've diffused enough out of the blind channel that they know that the not blind channel is the right way. And in this paper has got loads of citations from the people who say, oh man, cells have consciousness. Oh. And they write to me and they say, thank you so much for showing that cells have consciousness. And I say, 3 partial differential equations is enough to make this perfectly. So what you're saying is that consciousness is 3 partial differential equations or more.
Yeah.
And this is very funny because they don't understand what I'm saying. What the biologists used to say, and they don't anymore, we are winning. They used to say, yeah, this model's great, but real cells are complicated. They'd never do anything like this. So what I would say to a biological audience in the old days is at the beginning of the talk, I'd make people raise their hands as to whether they would believe evidence from a model if I showed them or not. And then, of course, you show them the real cells. And the real cells are perfect too. And I mean, here, in a sense, I'm cheating because I built this maze. I had this maze built by Michele, from the model, knowing what all the best parameters were and so forth. If you just go and arbitrarily build a maze, it might work, but it probably won't. But if you can model it and you know all the parameters, you can do it perfectly.
So these simulations show some really interesting things, namely that a large proportion of your population will not migrate at all.
That's right.
Yeah.
So this probably goes down to some of the questions we asked before, that there are multiple attractors and multiple mechanisms to get cells to where they need to be.
That's right. And do you see there is a difference? I mean, it's a good audience, this. You're seeing all the holes in my argument that I've had to patch up over time anyhow. Do you see in the model, everyone who is behind this wave is not moving at all. There's no directional information. Well, maybe those ones, but right. And you see in the reality, the cells behind do in fact have a little bit of directional information. So this is the thing I was talking about, David. The drug was not completely stopping the cells from signaling to one another. So what's happening here, it turns out, if the cells at the front are moving uniquely, but they are talking to the cells behind. And I'll flag up something to you that no one else would see in this. What should I do? Just keep talking and people can go off.
I think that's best, yeah.
Normally we go till 1:30.
Okay, I'll try and finish by then. I mean, you're asking such good questions, and I like answering them. So, okay. There are some PDA cells. I was trying to find the movie because I wanted to give you some, but I couldn't find it while traveling, I'm afraid. PDA cells, it will not surprise Sandy the tiniest bit. Don't solve the maze nearly as nicely. They're horrible, doing all sorts of other things, but they do solve the maze. There you see a PDA cell that finds its way to the outside. And the cancer cells do much better than randomly at solving these mazes. They're just not as beautiful as the blood cells or the big T cells.
What did you mean when you said the cells are talking to each other? Does that mean that they're creating an environment that they can get information from?
That is a very good question indeed. And I was eliding the fact that there's a whole load of stuff going on. So in the case that I showed you there, the cells that are migrating are secreting a chemoattractant themselves. But any way at all that the cells make a signal that can diffuse to the next one will give similar results. So breaking down the matrix would do fine. Breaking down the matrix is a great way of one cell talking to the ones behind it, because the one behind can see where the hole is. Secreting a repellent is a good thing. Using up the food is a good thing. The math that we use turns out to work with sort of antelopes and starlings and so forth. And starlings can actually literally see one starling away. They can just about see 2 starlings away. They can't see 3 starlings away. So all of the amazing patterns you see in clans of starlings are built on the starlings being able to see their immediate neighbor, and then the flock moves in an emergent way. Antelopes migrating across the Serengeti, it's just the same. But yes, you caught me. But cells talking to one another is a whole load of phenomena that you can express mathematically as related.
So you're saying that cells are conscious?
No. Go out with the person who is abusing the zoo. I've shown you these mazes. They're beautiful. They're absolutely wonderful. I love them. But are they biologically relevant? Yes, of course they are. When a tumor is metastasizing, it has basement membranes. The basement membranes may have holes in them. They may have made holes. One of the things I think is the strongest self-generated gradient in cancer is glioblastoma. And when I show glioblastoma, I'll show you one in a bit, people say, oh no, but we already know they move along the white matter tracts. That is plainly steering them. And the answer is the white matter tracts are nice holes where attractants could diffuse or repellents could diffuse. So it doesn't mean that the white matter tracts are necessarily making something specifically to attract glioblastoma cells. The fact that white matter tracts exist would make a self-generated gradient either go that way or go somewhere else. Other cancer types, if you have a cancer that is spreading by chemotaxis, which is certainly true of some, isn't true of others, but they must be using this sort of mechanism. It turns out that almost every chemotaxing cell is influencing the gradient that it's responding to.
But I have one other question, I'm sorry. All these videos are totally convincing, right? But it's cells migrating in near empty space. If we look at our skin layer, some of those are connected tissues, right? Could you comment on cell migration within a connected tissue?
That's really what this slide is probably saying in the tissue, there are probably a set number of gaps. In fact, that was going to be my comment. Oh, well, it could be embryonic development. When you're an embryo, the germ cells are actually made down at the distal end of your guts. Go figure why biology does that. But they must migrate by chemotaxis to your gonad or no babies. When the germ cells migrate, they are migrating through, so it's embryos, so the tissues are less well stuck together, but they have to find a path through cells that are in an epithelium and so forth. And the way they will do that is they will presumably use self-generated gradients to find where the best way is to go. Because wherever there is a hole in the epithelium, that is where the attractant will diffuse. And so that will be the way they're steered. Neural pathfinding, I've very little evidence for this, so this is purely hypothetical. But doing neural pathfinding with a self-generated gradient would be incredibly more efficient than the models that most people have, et cetera, et cetera, et cetera, et cetera. It's universal.
Yes.
I am going to show you here macrophages, bone marrow derived macrophages from a mouse's long bones. And this is them moving randomly in a chamber. And this is them moving randomly in a chamber which is homogeneously filled with the chemoattractant C5A. And you see the directionality of the ones that have been given C5A is astonishing. That directionality is not present in the experiment except where the cells are. The cells are making a gradient that is leading them in that astonishingly directional way. So immune cell, when you scratch yourself and so forth, the cells are making gradients. There are some dendritic cells in a maze. The dendritic cells do not form, there's the dendritic cells in another way, how nice. The dendritic cells do not just form a wave at the front with no one moving behind. So the dendritic cells are talking to one another. And we are working on what is actually going on in biological terms when the dendritic cells talk to one another. And it is going to make the most fantastic paper for the person who does it, because we know that the dendritic cells are communicating with one another, but the immunologists do not know that dendritic cells are communicating.
And they're leaving some trail, sort of, that others can follow.
It's like ants.
Content,
You can actually see, look at this. No one goes in here, right? Oh, damn, I cut it off. Sorry, this is the simple version. If you carry this movie on, and you see, if a cell randomly migrates in here, then suddenly all the other cells will come too.
Oh.
So that they are communicating. This is really nasty. So this is supposed to be an organoid. It's a really stupid term because it's just a ball of cells, but this is glioblastoma cells that have stuck to one another. And if you allow them to stick to a dish so that they can start migrating, then they migrate normal to the edge of they don't move randomly, they move in a directed way. Where does the direction come from? It's a self-generated gradient. How can I show that it's a self-generated gradient? Just imagine that I took a pump here and very slowly removed medium from here and pumped it back here. What would happen? I turn on the pump now.
Ooh.
And they don't do it.
What's the pump?
Takes away.
Yes, it's just recirculating the medium from, so it's all the same medium and so forth, but we just cause that to be a flow gently over the surface. So they can't keep a diffusional edge. This is all diffusion. Here's another thing. This paper, Julius Adler, you must know this paper from 1966. I was alive. I was only just alive. I think I was 9 months or something when this paper was done. So Julius Adler did this amazing picture showing that if you put E. coli on galactose agar, they form a ring by chemotaxis. And the interpretation from us now is that, which he's sort of got, Julius Adler's a very clever man. Inside this ring, there is no galactose. Outside that ring, there is saturating galactose. It's a self-generated gradient. And if you put them on complex agar, you get multiple rings. And it turns out that the first ring is serine, which is the best chemoattractant. So outside that ring, there is lots of serine, and inside there is zero. And then the cells that were left behind from that do aspartate, exactly the same thing. They like aspartate more, or have more receptors or something, and threonines before that.
Anyone ever use this as an assay to figure out what was the most attractive?
The most attractive turns out to be very hard to express mathematically. So it is a weird mix of how many receptors do you have, how well do the receptors couple to your migration, and how fast are you breaking down the ligand? Because if you're breaking it down faster, you get a steeper gradient.
So what I meant was if you had something complex, if you made a mixture, and let's say you couldn't break it out, you don't know all the things.
No, that's fine.
Use this as an assay to pull it out.
Yeah, it'd be interesting.
To back it up.
Yes, I think that lots of people have tried to do that.
Okay.
And they used simple explanations like this is more attractive and this is less attractive, as a result of which their experiments were useless. But if you revisited them and said, well, it's not that some things are more attractive and some are less, but that you have 3 PDEs talking to one another, then they'd find some quite interesting things built into their data. So I was made aware of this when I was speaking in a microbiology meeting, and someone told me about this paper. And so when I actually showed the slides, I showed this stuff in the microbiology meeting. And one of the people in the meeting said, do you know, Julius Adler is still alive and still has a lab in Wisconsin. Wow. And I phoned him up from that meeting and we had a nice conversation. He must be in his 90s now. I don't know if his lab is still running post-COVID, but it's a super story. Okay, why don't I try and run through a little bit, because your questions have been so good. Oh, but I have to show you this. This is King Henry VIII's maze in Hampton Court. And cells solve King Henry VIII's maze very much better than people do. If you see the real one of this maze, the people are close to random, whereas the cells, they're really quite good at it. But you see a really interesting thing, which is why I'm showing you. Do you see there's a glitch about there? Hopefully I've got a ring here, yes. Right, watch what happens. This maze was not perfectly made. And the cells know it wasn't perfectly made, and they squeeze through. So what this glitch, this is the equivalent of the door in IKEA. The older ones of you, especially the ones with children, know you have to go to IKEA, and there is a little door, and if you go through the little door, you don't have to look at all the sofas. And it's fantastic. You save like 2 hours of your life. And these cells are missing the sofas in IKEA. We like this so much that we built a maze where we built a real shortcut there. And the answer is both mathematical and anthropomorphic. So there are the cells solving King Henry VIII's maze. And they are going to be able to tell that there's a shortcut. And some of the cells take the shortcut. Right. Now, I've told you what's going on here. Those cells at the front are breaking down all the chemoattractant and making a gradient, which means that these cells here can't have any chemoattractant because the cells in the front broke it all. So, these cells can tell everything about their environment by breaking down a chemoattractant. I mean, it's mad, except that it's what's going on in your bodies. And the second thing, which is the anthropomorphic thing, is that those cells which didn't take the shortcut are condemned to spend the rest of their existences in the sofa section of IKEA because the ones in front have taken all the information away.
Basically like sabotage.
It's really, well, it's competition. Yes, you're talking about collective migration. The ones in the front are doing collective migration, but the ones behind are stuff them. Gosh, this is fun to talk about, but I want to talk about one more thing before we go.
Are those mazes 3D printed or?
Yes. Well, no, sorry. We have failed to 3D print them. We etched them. We are in the middle of trying to 3D print them, but the details are not quite fine enough to do it nicely. Okay, I showed you this thing at the beginning, and I want to answer it before I let you go away. So, all of the attractants that I've shown you are getting broken down. They're getting destroyed. LPA is destroyed by an enzyme that takes the phosphate off LPA and just turns it from being this lipid with a phosphate head group to just being a lipid. So it goes from being a strong chemoattractant to being nothing at all. But what happens if you take a chemokine and you chop it with an enzyme, and so you turn it from being a strong agonist to a weak agonist, meaning that if 100 receptors are bound to this agonist, 10 of them are put in an active state and 90 of them are held in an inactive state. So that weak agonist, if you measure chemotaxis, cells are still going to chemotax to it. It's still an agonist. There are other enzymes that destroy chemokines, like cathepsins. Okay, so I'm not talking hypothetically. I am interested always from a mathematical and theoretical viewpoint, but this is totally real. Every chemokine is cut by several different enzymes, and very often it is known that by cutting it with these enzymes, you change its properties. Okay, so if you have a gradient, and this is answering some of the questions earlier in a figure, if you have a linear gradient of a chemokine, for example, and you look at the receptor occupancies, they get more and more saturated as you get higher and higher, so they get less and less potent. And the thing that gives you chemotaxis is the gradient of this receptor occupancy, so D occupancy by dx, sorry. And what you are seeing when you see chemotaxis is this. And the answer is in Sally-Zygmund's assays, the cells all move. But what you should see is that the cells at the high end of the chemoattractant move slowly and the cells at the low end move efficiently. So anywhere where you see all the cells moving, it's not simple.
Yeah.
It's brilliant. Right, saying you have an antagonist. So a thing that binds to the receptors but does not activate them. So if you have a constant amount of attractant there in green and an increasing concentration of an antagonist, then your gradient of occupied receptors does the same thing, but the activated receptors now goes the other way, it goes down. So actually the gradient in activated receptors now points the other way. The D occupancy by dx now points towards me rather than away from me. Does this tell you anything about reality? Well, here's some cells given a constant gradient of a chemo. Oh, stop it. Here's cells given a constant flat non-gradient concentration of an attractive chemical. They like it, they migrate, but they don't go anywhere, there's no gradient. Here it is an antagonist. This is a chemical that binds to the receptor but does not activate it. And you see the cells don't like it. And there's no information because there's no active receptors, so they don't go anywhere. But if you mix these two gradients, the cells come backwards. And this is the best movie of negative chemotaxis that has ever been taken. I mean, the most efficient backward chemotaxis. So the model is quantitatively telling us how the cells are going to behave. We're in a lovely place now. And I think probably more than half of all the times when people report negative chemotaxis, in other words, things chemotaxing away from a chemical, I think that more than half of the times it's going to turn out to be this. It's going to turn out to be an interaction between a positive chemoattractant and something that's blocking it. We're going to ask a question. Oh, no.
I've got a question. So obviously the cells are a certain dimension, and you've got what you showed the gradient there was, I don't know what the dimensionality of it was, but is really a temporal dynamic than a spatial
You want to do a sabbatical in the lab? You're invited.
Of course.
I think I know how to test that. I think it's complex. My entry into this field came trying to answer a question like that. But the answer was so full of PDEs that none of the biologists have ever paid any attention. And I was not yet very good at bridging the gap between the two. But I think it is an emergent property of the cell perimeter moving. And I think it is actually probably purely temporal. But the cells are moving, so a temporal gradient correlatively becomes a spatial gradient inevitably.
The frame, if you move the frame, it's effectively spatial.
That's right. But I mean, if you're in a gradient and you stick out a pseudopod, this was the answer with the single cell self-chemotaxing. If you move a pseudopod, then the receptors in that pseudopod are going up the gradient that you yourself have created, whereas the cells at the back that are not on a pseudopod are not. So there, by moving spatially, you create a temporal gradient. Okay, I'm going to make it. If the attractant is degradable, then you get a complex behavior. And this is predicted by analytical math. There is a nice analytical math heat map. I would have talked for 10 minutes if I had 10 minutes, but I don't. What you can see is the dead bit in the middle is a function of the concentration of attractant and a function of the concentration of the inhibitor, and you can work out the parameters by where the dead point goes. If the repellent is degradable, you get a really cool emergent behavior. And I'm just showing it for the fun of it here rather than going to explain it all to you. But again, the analytical math, read the paper if you want to see.
Amazing.
Isn't it nice?
Yeah.
And actually you can see that one edge is kinetically stable and one edge is kinetically unstable, which is also. And again, the place that is there is nothing here apart from the midpoint of a system of equations.
That's as far as you can get.
And it moves as the cells break down more and more attractant, so more and more repellent. So this thing in the middle will, if you keep watching it, slowly move towards the high end as they break down more and more of what was put in. Okay, got to the end of the talk. It's going to be great. Saying you have a weak agonist, so one that only activates some of the receptors. So saying we have this gradient of a weak agonist, the gradient of occupancy of the receptors is going to look like that. So if you put cells in this weak agonist, they're going to migrate that way, up to the occupancy. Now we've got a gradient of a strong agonist, but we've started it at a non-zero place. And again, the occupancy gradient, I'm giving the punchline away too early, but I just caught it. So again, the gradient of occupancy goes that way. So again, the cells will go up this gradient. But when we mix them, the gradient of occupied receptors goes up here. This is the proportion that is with the weak agonist, and this is the proportion of the strong agonist. So if we go and render the fraction that is stimulated of the receptors, the gradient is now coming this way, even though both of these gradients are attractants. And both of these will send the cells that way on their own, but when you mix them, the analytical math is telling you they're going to come this way, and I showed you the result. The analytical math is right. One gradient, two gradients, mixed gradient. And we can quantitate it all, and the quantitation makes it even clearer because your eyes are better at seeing the special case. There is the strong attractant, and you see most of the cells are going up the gradient of it. And there is, this is, I was talking about coding, I wrote the code for this in a flight to the US. There is the weak attractant, and there is the mixture. Isn't that lovely? And you see, it was no surprise to us because we've been doing this long enough that we knew our models were probably going to tell us what we, and if we didn't get this result, we'd have also been very happy because we'd have known there's one parameter we had to go and chase. Okay, so weak chemoattractants are attractants on their own. They become repellents or other complicated things in the presence of other ones. And you have to investigate competition. And if you were looking at what steers cancer cells, all cancer cells are being steered by multiple things. So you have to be aware that the results are interesting. And my general conclusions are complexity is wonderful. Complexity is very hard to study biologically, and biologists get confused in their heads, by which I mean our heads, because I am a biologist, even if I'm a bit of a convert, which is to say that if you have too much complexity, you can't measure something reproducibly. So biologists tend to say, okay, we've got to iron all the complexity out of our experiments and get something simple. But simple experiments aren't interesting. Complexity is what makes biology and medicine and cancer and et cetera interesting. And that complexity is your friend. So cells and attractants interact with one another. And cells and adhesion molecules interact with one another, and the same thing has been shown with adhesion molecules and tension. And the information that makes you interesting, and probably makes cancer spread too, comes from this interaction. So don't wipe it out. And these interactions give you non-trivial results, and the non-trivial results are what you want.
Point.
And I made it to the end. Thank you very much indeed. Math PhD, theoretical physics PhD. Didn't like experiments and is now a statistical consultant. Did like experiments and is now the best microscopist in the lab.
Nice.
Yeah. Oops.
Great.
Thank you.
Yes, thank you.
So we're already over, but any last questions for Rob?
Yeah, you showed some of your immune cells in your maze.
Yes.
Clearly convincing that they generate their own
Yes.
radiance. But we also know that they have to follow some cues to get to the site where
Yes.
they need to be. So do you see any nonlinear combinations of attractant versus self-produced attractants?
When you have asked nonlinear, if a cell is responding to a cue and is also breaking down the cue while it responds, in one dimension, that changes a linear stimulus to almost an exponential stimulus, which is much easier for the cell to read. And actually, a lot of chemoattractants which can't be broken down are listed in the literature as being weak chemoattractants, even though they are in fact strong chemoattractants. But they're weak chemoattractants because the cells can't make the gradients exponential. They just work as linear, and that gives you less information. The question you asked, can the interactions give nonlinear results as a consequence? I mean, the amazing thing is I was lucky enough to have been working in dictyostelium in my PhD. So I knew I could find cells that only had one or two things that they were responding to. We are now finding, so the dendritic cells that I showed you earlier, probably responding to about 3 chemoattractants. So I am going over the next 5 years, if people will put up with me and keep doing the experiments and the math, I will be able to get good analytical models for dendritic cells, I think. Macrophages, but you can dominate macrophages, but macrophages are very nonlinear. Neutrophils, my mission is to persuade people that they shouldn't be studying quantitative effects in neutrophils because they don't understand the quantitative behavior at all. They're very interesting and very important, and we should be getting there. But don't try and make a model of neutrophils because you will fool yourself.
Thank you.
Well, I have a quick question. One of the things that you said repeatedly was about the kind of receptor saturation, but receptors are dynamic, and so they can desaturate. Do we understand the timescales of that? Because one would imagine that could be important for gradient sensing.
Yeah,
right?
good question. So there are two answers to that question. One answer is that there is no receptor modification in our models of dictyostelium, and they're as near to perfect as damn it. There is really essentially no difference between what real cells do and what the computational and analytical models say they will. So in those dictyostelium, desensitizing the receptors and synthesizing more receptors and so forth is not relevant. In any other biological system, you have to answer it question by question. Dendritic cells, it was published, in fact, DCs, no, it was neutrophils. It was just published last year that the enzymes that modify the receptor affinity are totally important in being able to respond over a field. Case by case. And mercifully, receptor desensitization is really easy to model. So anytime we want to, we can shove it in a model. You just add a couple more lines of code, and we can see.
Yeah.
But I haven't needed to yet, fortunately.
One minor point is to let everyone know that you've published quite a lot of this work.
Published.
And what paper would people go to?
Okay. There is a science paper with the mazes in. And the science paper says a whole load of stuff that I haven't talked about at all, about how important diffusion is. Because most work that people do on chemotaxis, amazingly, does not consider the diffusion of the molecules. And that paper is talking a lot about it. There's a current biology paper about a month ago which has got all the reverse chemotaxis. The current biology paper is my favorite paper. It's liquid, and hopefully I'll get around to writing a thing for your blog, or David, about it.
You also have a Quantas article. Is this something that maybe high schoolers in the audience might be interested in?
Yeah. There are quite a lot of, actually, there's a Scientific American article about it, which was, so Scientific American, or what's the name of that?
Quantas?
Quanta, yeah. There's quite if you just look up Insall and Mazes, or Tweedy, who's the first author in Mazes, you can see a decent number of pieces of all sorts of different levels on it. But actually, the current biology paper is relatively clear and relatively nice, so that would be good. And there are 2 current opinion reviews, one that just came out and one where Tweedy is the first author, and the Tweedy one is the best.
It's good to know priorities. Thanks, Robert.
Right, well, thanks again, Rob, very nice.
So technically you have lunch now, right?
Last day of science.